Good evening, Professor Majumder. Can you hear me? I think you're muted. You have to unmute yourself. It's on the bottom.
Yes, I'm able to hear you.
Okay, thank you. So we'll start around 7 p.m. and then we'll go ahead with
That's
that.
absolutely fine. Are you able to hear me well?
Yeah, I can hear you perfectly fine.
Okay, all right. Thank you.
Thank you. Thank you.
Hello, good evening, good evening.
I think it's on time. Welcome to the Progress and Prospects in Biology webinar series. Thank you once again for joining. We truly appreciate your support and kind cooperation to host the webinar series for the last 6 weeks. And I'm delighted to say we have more than 1,300 subscribers who showed interest to join the webinar series. And approximately we receive around 400 to 500 viewers on average for all the talks. A couple of housekeeping stuffs before we start today's webinar. Please mute your microphone, turn off your video, and don't share your screen during the webinar. Please refrain from putting any welcome message in the chat box and post your questions for the speaker, and we will try to answer them at the end of the session. Those who require e-certificates, a feedback link will be shared at the end of the session in both Google Meet and YouTube. However, please note we'll not be sharing the link after the session is over. With that note, I now request Professor NRI Banerjee, a professor in zoology at the University of Calcutta and the mentor and convener of the webinar series, to say a few words and introduce today's speaker. Thank you.
Thank you, Shaunak. Very warm welcome to all the participants who will be joining us through this Google Meet platform as well as YouTube, and all the future participants who will be doubtless tuning in to listen to Professor Majumdar's archive talk in our YouTube channel. Progress and Prospects in Biology is a platform that is very, very young in its concept, but we are using and I again request the participants to please mute yourselves and also turn off the video to preserve the bandwidth. So Progress and Prospects in Biology is a virtual platform put together to discuss science. Today we are very fortunate and honored to welcome Professor Partha Pratim Majumder to talk about what is doubtlessly on our minds all the time, round the clock, and the terror of COVID-19, SARS-CoV-2, the virus, what it does. In one of our previous talks, Professor Dhrupajyoti Chattopadhyay has told us about the virus and the drug discovery efforts about the virus. Then another Dhrupajyoti Chatterjee, who has come up with a paper-based assay to detect the virus, also spoke. And today we shall listen to Professor Partha Pratim Majumder on the evolution of SARS-CoV-2 virus and doubtless other things as well. So I welcome Professor Majumder for sparing the time for us, and it is my honor and privilege to introduce. He does not need any introduction, but nevertheless, this is a formality. So here goes. Professor Partha Pratim Majumder is the president of the Indian Academy of Sciences since 2019. He is a distinguished professor at the National Institute of Biomedical Genomics, Kalyani, as well as a professor at the Indian Statistical Institute, Kolkata. He was the founder director of the National Institute of Biomedical Genomics from 2010 to 2016. He was a director there. He was selected as the National Science Chair by the Government of India in 2020. Hearty congratulations for that, Professor Majumder. He did his PhD from the Indian Statistical Institute, Kolkata. His research involves the study of variations amongst genomes of individuals who are often clustered in social groups in geographically proximal regions to draw inferences on human evolution, population structure, susceptibilities to diseases, responses to drugs and vaccines. He's also interested in statistical applications and in developing methodologies for drawing statistically valid inferences from genomic data. But I would be failing in my duties if I did not mention the enormous influence he wields over the scientific community in general, and an educationist much loved by his students. So thank you once again for joining us, Professor Majumder, and over to you.
Thank you, Professor Reproductive. It's really a pleasure. I will start with a note of gratitude and also a note of apology. The note of gratitude is that all of you have invited me and all of you who have assembled here to listen to me. So that's really, I'm very, very grateful to you for doing that. My note of apology is the following, that a couple of days back, I was in e-conversation with Professor Banerjee, and she mentioned to me that a lot of people have signed up and my audience is very heterogeneous. So the apology is that I will not be able to satisfy any of you, as a matter of fact, and that's my note of apology. The reason being that in my talks, I try not to lose anybody completely. So what I'm going to do is that I'm going to pitch my talk in such a way that everybody will derive something out of my talk because of the heterogeneity starting from undergraduates to even professors. So the way that I will pitch my talk is that everybody will have something to understand from my talk and hopefully learn from my talk. So sorry that I cannot be completely satisfying you completely through my talk, but I hope that you will listen to me and be able to gain something out of my talk. So I'm going to talk about evolution of the SARS-CoV-2 virus that has created a panic and continues to create a panic in not only in India, but throughout the world. It's become a pandemic. Started as an outbreak in Wuhan in China, and then the outbreak became an epidemic, and then it quickly became a pandemic. And the rise from the outbreak to becoming a pandemic was really extremely fast. So that's very unusual that we had in about a month or 1.5 months time an outbreak becoming a pandemic, where the World Health Organization declares the infection to have become a pandemic. What I want to do is, from an outbreak to become a pandemic, the virus obviously has spread geographically. So I'm going to talk a little bit about the spread of this virus. And secondly, what I'm going to do is also talk about the evolution of the virus. So I'm actually going to split my talk in 2 or 3 parts. In the first part, I'll talk about the characteristics of the virus, a little bit about the evolution of the virus. The second is what, tying with the evolution, what gives it some kind of a selective advantage for it to spread across the world so quickly. And then I will present to you an enigma, an observation that really raised curiosity in our minds, and been able to solve that curiosity to a certain extent. Can't say that we have solved it completely, but we have gotten some insights. So that will be the third part of my story. Time permitting, the fourth part of my story will be on 2 vaccines that seem to be near the door. So I'm going to talk about those 2 vaccines. And that will be the end of my talk. So all of you know that these viruses are very, very strange. They are living and non-living. They are living in ways that they have certain properties that all living beings share. And they are non-living in the way that they actually cannot reproduce by themselves. They do not have the appropriate machinery to reproduce by themselves. They need a host. So without a host cell into which they would enter, they cannot reproduce themselves. So they cannot make copies of themselves unless they have a host cell in which they enter. And that's the reason they are often called as non-living entities. They contain nucleic acid. This particular virus that we will be talking about, the SARS-CoV-2 virus, is an RNA virus. And I will qualify that RNA by saying that it is a positive stranded RNA virus, and that positive stranded RNA virus gives it some kind of advantage already, and I'll come to that in a minute. It's a positive stranded RNA virus, and it's one of the longest viruses that we know. Its genome size, the total number of nucleotides in the RNA, is 30,000. So it's one of the largest RNA viruses that we know. It's called a coronavirus because, as you see, it's like it has these flares, the spike proteins. These spike proteins are important for it to go and attach itself to the host cell. And I'll talk a little bit more about the spike protein. So it's a coronavirus. It looks like a sun. It looks the spike proteins look like flares of the sun, and that's the reason why it's called a coronavirus. This is not the only coronavirus that's known to infect humans. There are other coronaviruses, not too many of them, but there are other coronaviruses that infect humans, and we have met some of them already. And I'll describe the ones that we have met, primarily because they have created some problems in us. Just like the SARS-CoV-2 is creating some problems, clinical problems in us. There are other coronaviruses that infect us but don't really create any clinical problems, and so we call them as benign, and we tend to ignore them. At the same time, they also elicit immune response, so it's not like we can completely ignore them. So this particular coronavirus belongs to this entire family of coronaviruses. It has a large genome, and of course it's wrapped in protein. It's actually wrapped in lipids, and that's the reason why we are asked to wash our hands every so often, because what soap does is to essentially remove the lipids and expose the naked RNA, which then essentially gets sheared and degrades. And so the virus is dead. It can't go and infect the host cell, the human cell. So that's the reason why we are asked to wash our hands every so often. So this particular virus, so let me talk about the evolution of this particular virus. This virus, we believe, came to us, crossed a species boundary, came to us from the bats. And the bats actually harbor a very large number of viruses. We don't really know why they harbor so many viruses, and they themselves don't get killed. There are speculations. I'm not going to provide you with the speculations because it's irrelevant to our discourse today. Bats are home to a large number of viruses, including many coronaviruses, and it's believed that this particular coronavirus, SARS-CoV-2, also descended on us from the bats. How do we know what is ancestral and what is descendant, and how a virus has evolved? So we collect coronaviruses wherever we can, and we sequence them. And so we determine the RNA sequence of each of these virus entities or viruses, and then do what's called a phylogenetic analysis of these viruses. What phylogenetic analysis does is it essentially does pairwise comparison of sequences of these various viruses, and then puts these sequences into clusters. And this clustering is done by comparing base by base the sequences and finding out which of these sequences are most close to each other. And if they are very close to each other within a certain threshold of difference, then we put them in the same cluster. And so we do a phylogenetic analysis that provides us with relationships among these sequences that the viruses have. This particular, it's like a tree. It has a root, it has branches. So we can actually root the tree. Again, I do not want to get technical because phylogenetic analysis itself is a technical pursuit, but it ultimately it's very informative. It ultimately tells us that what is among these viruses, which one is the root of the tree and which one are branches. By that I mean the root of the tree is essentially the ancestral virus, and the branches are the descendant viruses. And this is all done by comparing the nucleotide sequences of if it's a DNA virus of the DNA, if it's an RNA virus of the RNA sequences. And of course, you can imagine that those that are very close to each other, the differences are small. They likely belong to the same ancestor or descendants of the same common ancestor. In this particular case, like I said, that this coronavirus arose and the outbreak came from Wuhan, and the person who got infected, and she was dealing with seafood market. She was a worker in the seafood market, and in the seafood market, they also deal with various other kinds of animals in addition to seafood. And she came down with some clinical symptoms, and they identified that she was infected with the virus. They sequenced the virus. Incidentally, that worker in the seafood market is still living and did not die of the viral infection. So they took that virus, they took the sequence and found out that it's a coronavirus. And then they looked at all of the other coronavirus sequences that were known and found out that it's very close to the bat coronavirus, and the sequence identity between the bat coronavirus and the human SARS-CoV-2 is 96%. So that means that of these 30,000 nucleotides, 30,000 alphabets that there are, 96% of those alphabets are exactly the same, are identical between the bat coronavirus and the human SARS-CoV-2. There was also a speculation that did it come directly from bats to humans? Did it cross the species boundary directly from bats to humans, or was there an intermediate species and the bats infected that intermediate species, and then that intermediate species infected the humans? And the reason why one felt like that is, again, the bats are not something that's dealt with in the seafood market. We do come in close contact with the bats, especially when trees are being felled, and in the construction sites, etc., we do find bat colonies. But the speculation was that there may have been some other animal species with whom we come in closer contact, especially in seafood markets, etc., that may have been an intermediate species. So from the bats, intermediate to this particular species, and then from that intermediate species, there was crossing of species boundary, and we became infected. What may have been this intermediate species? It was serendipity. Accidentally, some discovery was made, and that led to widespread speculation. I will still call it a speculation because we don't really know whether exactly that happened. So what was found about 150 kilometers away from Wuhan, they found two pangolins. Pangolins as these scaly anteaters. They found two pangolins that were dead with a lot of froth in the mouth, etc. So whoever found them, they speculated that these two pangolins may have their lungs infected. Otherwise, why would they have so much of froth in their mouth and be dead? So they actually did an autopsy of the pangolins. They collected the lung tissue, and lo and behold, they found that it was infected with the virus, and it was infected with the coronavirus. Then they did the obvious thing. They sequenced the coronavirus, and indeed they found that it was the sequence identity was very close to the human coronavirus, also close to the bat coronavirus. So the jury is still out whether the pangolin was an intermediate animal species, but most likely the pangolin was an intermediate animal species, and pangolins are also hunted, killed, and eaten in many parts of the world. So we believe that from the bat, it came to the pangolin, and the pangolin infected the human. So that's really how the evolution took place. I said that this is a positive stranded RNA virus, and that is important. It's positive stranded RNA virus. That means that it is like an mRNA, already ready to be translated. So the transcription step is skipped, and you're already ready to make it into a protein. So it's like an mRNA. And this particular fact has been exploited by, if I have the time and talk about the vaccine, this particular fact has been exploited by one of the companies that's trying to make a vaccine. So this is a positively stranded RNA virus, which means that it is very, very easy to translate this, and that's one step ahead in terms of its ability to spread and infect. This coronavirus, so we'll come back to this positive stranded business a little later, perhaps towards the end of my talk. This particular coronavirus, as a matter of fact, is very kind, and I'm saying this with some data. It's not speculation. So if we look at other previous outbreaks or epidemics of coronavirus, actually there were no epidemics. There were only outbreaks of previous outbreaks of coronavirus. So SARS-CoV, one didn't expect that we would have CoV-2. So it was not named as CoV-1, it was just named as SARS-CoV. Also arose in Wuhan. That was another coronavirus that was named as SARS-CoV. And if that particular coronavirus arose in Wuhan, there was an outbreak, but it was contained, and it didn't spread very much out of Wuhan. That coronavirus killed, of the 100 people that it infected, it killed 11. So the killing power was 11%, which means that it was quite virulent. It was 11 out of every 100 people on an average that it infected, it killed. If you look at another, so this was in 2003.
I think we are facing some technical problems from Professor Majumder. We'll try to fix that as soon as possible.
Am I audible now?
Yes, sir, you are audible.
So what has happened is that suddenly the electricity went away, and therefore I'm doing this from home. The electricity went away, and so I lost my Wi-Fi, the router also. Anyways, now I'm back.
No problem, sir. No problem.
Yeah, so now I'm back, and I can continue. So what I was saying is that this happened in 2003 when SARS-CoV outbreak took place in Wuhan, and 10 years later, so that killed about 11% of people that it infected, and later about 10 years later, in 2013, there was another outbreak, and that outbreak was took place in just give me a second. That outbreak took place in the Middle East, and that's called the Middle East coronavirus. So the Middle East coronavirus was even more dangerous. It killed about 34% of people that it infected. So of the 100 people that it infected, it killed about 34 on an average. So that was a more dangerous virus compared to the SARS-CoV, and that's called the MERS coronavirus. So we have SARS-CoV, which is 11%. We have the MERS, which is 34%. And if you compare that to SARS-CoV-2, which is what has spread and which is actually scaring us now, the overall, on an average, the percent of people who are killed is 2 to 3%. So of the 100 people that it is infecting, only 2 or 3 are dying. So I call this virus as a kind virus, and that's the reason why the SARS-CoV-2 is a kind coronavirus. Now, a virus actually does not want to kill its host, because if it kills its host, it also dies, and therefore it is not to its advantage to kill the host. So it actually wants to infect the host, and it wants to reproduce within the host cell, but actually does not want to kill the host. So this is the reason why this particular coronavirus that is not killing as many like the SARS-CoV or SARS-CoV-2, SARS-CoV or MERS is able to spread so well and quickly became an outbreak to an epidemic to a pandemic. Is this the only reason why it has been able to spread so well? So we actually were collecting data. It's curiosity. I normally don't work on infectious diseases, but this infectious disease was something that all of us have been interested in. One from fear, the other is from awe. That why is it that it is spreading so much? We've never seen a pandemic in our lives. The last pandemic was the Spanish flu, which was in 1918. None of us was born at that time. So this is the first time that we are actually encountering a pandemic. So when we were collecting data, unfortunately, two things were happening. The epidemiological data pertaining to how many people are getting infected, how many people are getting killed, and all of that. Those were being put on a public domain database. Also, as these sequencing projects were being undertaken in larger numbers in various parts of the world, the sequences were also being put on a website called GISAID or Next Train, which is a sister or a subsidiary website. So we were able to catch hold of or get access to both epidemiological data and sequence data, and that's quite interesting that we were able to do it. When we looked at the epidemiological data and we compared the epidemiological data with the viruses, with the sequences of the viruses, the one thing that panned out, of course, is that it is spreading very fast. It's not killing as many. And then when we superimpose that on sequences, what we found was that the virus was also evolving. Of course, viruses mutate and the virus was evolving. Fortunately, this particular virus was not evolving as fast as, for example, the influenza virus. It's evolving at a much, much slower pace. And the reason why its rate of evolution is so much smaller, orders of magnitude smaller, is because this virus has the proofreading ability. It has the appropriate enzymes to proofread, and therefore, anytime there is a mistake in copying of the RNA, it actually corrects it. The proofreading enzymes actually make the corrections, and therefore the mutation rate is kind of low. It's orders of magnitude lower. So the virus is evolving not very fast, which is good, not very fast, and which is good from a vaccine or drug development point of view, but we'll come to that a little later. What we were able to then do is to look at these sequences and again do a phylogenetic analysis. And not just us, people around the world were doing these kinds of phylogenetic analysis, trying to find out how to relate various subtypes of the virus with their mutations and so on. And what we quickly discovered is that in about a month and a half, the virus had evolved into 11 different subtypes, including the ancestral. The ancestral subtype was in Wuhan, is rooted in Wuhan, and that ancestral subtype is called O subtype, and it evolved into various other subtypes, which scientists, virologists gave them names such as A2, A2A, B, B1, and so on and so forth. So anyways, the names don't matter. They're including the ancestral. In about a month and a half, 2 months' time, there were different subtypes that evolved of this particular virus. Now, when we looked at the spread of these various subtypes, something astonishing struck us. What was astonishing is that what would one expect that there are 11 subtypes. Most of these subtypes would be available in most parts of the world because the virus is spreading irrespective of what subtype it belongs, unless some subtype had some kind of an advantage, and then it would dominate the other subtypes. It would outcompete the spread of the other subtypes and then spread. The astonishing part was just this, that instead of even if 11 different subtypes didn't populate all parts of the world, we would expect 5 or 6 different subtypes to populate every part of the world. And that didn't happen. What happened was that there was only one subtype, the subtype called A2A, which completely dominated and outcompeted the other subtypes and dominated every geographical space. This was extremely surprising because unless the A2A subtype had some kind of selective advantage in a Darwinian sense, one wouldn't expect this to happen. And this is when we got interested, and this observation we were looking at, came from a former PhD student of mine, now a colleague, and then we started looking at the sequences and so on and so forth. His name is Nidhan Biswas. I'm telling you the name because many of you actually may know him, and he needs to get credit for the observation that he made, and subsequent to that, we started doing this analysis. So we found that this subtype A2A actually spread throughout the world, and then we started looking at the mutations that had accumulated in A2A compared to the ancestral type. And of the mutations of the various mutations, not too many of them, of the various mutations, one particular mutation stood out, which is near the spike protein, like I said, there are these spikes that attach itself to the host cell, and I'll come to that attachment in a minute. With that spike protein, there is a specific mutation that changes an amino acid from aspartic acid to glycine. So it's at position 614, so it's called D614G mutation. So this is a non-canonical mutation. It changes an amino acid, aspartic acid, to glycine. And what this does is it provides us this particular subtype of the virus a huge advantage, and that advantage I'll tell you in a second. Before that, let me explain to you what the spike protein is all about. The spike protein is usually denoted as S. It has two components. One is called S1, the other has been denoted as S2. And this is joined by a hinge. And unless so the S1 is the portion that goes and attaches itself to the human cell, and it attaches itself to a specific protein called angiotensin converting enzyme 2. ACE2 is where it goes and attaches itself, this S1 subunit or component of the spike protein. The ACE2 protein is abundant in lung cells of a specific kind called pneumocytes. So these lung pneumocytes have huge quantities of ACE2 expressed on the surface, and that's the reason why the virus is able to go and attach itself to the pneumocytes and be able to anchor there. But anchoring doesn't help. It's still outside of the cell. It needs to get inside of the host cell to be able to do that, and that is enabled or facilitated by the S2 component of the spike protein. But like I said, between S1 and S2, there is a hinge, and that needs to be cut so that these two can come out, these two can separate, and the two components can act independently. So the S2 protein can act independently. The S2 protein is like a pair of scissors that will let the virus enter the cell. This particular subtype A2A is able to anchor itself much better because the glycine is able to anchor itself, is attach itself much better to the ACE2 protein. So it anchors better, but does it also enter better? We don't know that. Then we started looking at what helps the entry into the host cell. So it's essentially the fusion of the two membranes, the viral membrane, viral capsid protein, and the host membrane. Those two will fuse and then it will enter. But there has to be this shearing of the cell membrane. And that's done by a serine protease, and that serine protease is called TMPRSS2. The TMPRSS2 is a serine protease that actually helps the virus enter the host cell. And then once the virus enters the host cell, then of course it will use the host cell machinery to reproduce itself, make multiple copies, and then go and infect another individual, and so on and so forth. So that's how it does. So we looked at TMPRSS2, and we found that it's not just TMPRSS2, but there are also other players that are involved there, interferons and so on and so forth. I'll simplify the complex story a little bit, but so that's how it spreads, and that's how the A2A protein is able to spread so well, has a selective advantage in attaching, and then something happens, and I'll tell you in a minute. How do we know? So it's spreading, and how do we know? A question is oftentimes asked, and we ask this question every day, or you are asked this question every day. I'm asked this question every day by laymen, that when is this damn thing going to stop? I mean, the spread going to stop? So we can give our subjective reply, and it's going to be subjective, but we need an objective measure to be able to see whether whether the virus is still spreading or whether it's coming down. What is the objective measure? And epidemiologists have various kinds of objective measures. And one objective measure is doubling time. So if you looked at the Telegraph 2 or 3 days ago, and I'm talking about in a global sense, you would see that globally, from 1 lakh people infected to the next 1 lakh people infected, the doubling of the total number of infected individuals took about 18 days. Today, it's taking about 3 days. So it's spreading, it's still spreading very fast. When this doubling time elongates, when doubling time becomes larger, then of course you know that the spread of the virus is becoming lower. The another objective measure is what's called R naught. R naught is a measure. It's called the basic reproductive rate. It essentially tells us how many uninfected people is one infected people able to is what R naught is. And you can imagine that if R naught is large, 5 or 6, and uninfected person is infecting 5 or 6 people, then those 5 or 6 infected people, each one of them will go and infect another 5 or 6 uninfected people, and so it's spreading very fast. If one infected person can infect only one uninfected person are not equal to 1, then you know that it has stabilized. And it starts to reduce when R naught becomes less than 1, when one infected person is able to infect less than one uninfected person. That's when you would say that the spread of the virus is under control and the spread of the virus is coming down. So this is interesting. So the question is, how do we estimate R naught? So R naught actually has 3 components. 2 of them are not under our control. The third one is under our control, which is the third component is the number of susceptibles in the population. Number of uninfected individuals in the population who can be infected. Now, that's the total number, that's the catchment of the total number of susceptibles that an infected person can actually go and infect. If we can bring down the number of susceptibles, then we are in business, because then R naught will be under control. How do we bring down the number of susceptibles? Is that under our control? Of course, it's under our control if we can find a vaccine. So what's the vaccine? That is really a proxy virus. The virus elicits various kinds of immune responses, and if you have a proxy virus, that will also elicit the similar immune responses, and that immune response will prevent the true virus, the real virus, from infecting an uninfected individual. So if you're able to vaccinate a large number of uninfected individuals, then you actually bring down the pool of susceptibles to a small number, and therefore any infected person in the population will not find many people to infect, and that's how you control an epidemic when a vaccine is available. Unfortunately, we are able to control this epidemic because the vaccine is not there, and that's the reason why there is huge hue and cry about finding a vaccine all over the world. Okay, so I've talked about evolution, I've talked about spread of the virus, I've talked a little bit about the biology, and now I'll tell you about another observation that we made and that brought us into deeper biology, and we are able to understand how this virus is able to even enter the host cell. So like I said, that attaching itself to the host cell doesn't do the entire job. It needs to get in the host cell. The protein Empress2 with detachment or distinction of the S1 and S components of the spike protein. The S2 component of the spike protein will allow the virus to enter the host cell. What was the observation that we made? Well, I told you one observation, which is that this particular subtype of the virus, the A2A subtype, has been spreading mercilessly throughout the world. Good. So we looked at now the various subtypes in various continents, and to our surprise, what we found was that in East and South Asia, the virus A2A was again dominant virus, was the dominant subtype, but there were also other subtypes that were still existent in East Asia and Southeast Asia. While in Europe and North America, all of the subtypes were actually gone. This particular subtype, the A2A subtype, outcompeted other subtypes and completely spread itself so well that that's the only subtype that there was no diversity, or there is hardly any diversity in the viral population in Europe and North America, while there is some amount of diversity in East Asia. So we said that why is it that this particular virus is not able to spread as efficiently in East Asia and South Asia? And we postulated a hypothesis. I'm a human geneticist. I work on humans primarily. And we also know that there is, host viral, host bacterial, host pathogen interaction. So we postulated that the ancestry of East Asian populations is quite different from the human populations. Now I'm talking about the ancestry of East Asian human populations is quite different from the ancestry of European or American populations. The East Asian, again, this is not a discourse on human genome diversity or human ancestry. Suffice it to say that the ancestral component in East Asia is quite different from the ancestral components of humans in Europe and America. So what we thought was that let's look at the human genome, let's look at the human genomes and let's look at those components of the human genome that the virus uses. And of course, one of the best, one of the most prominent genes that virus uses is ACE2. We looked at ACE2 and it actually didn't help because with respect to ACE2, there is very little difference across the world. So you don't have any polymorphic loci in ACE2 that could be attributed to its success of the virus in spreading and the differential success between East Asia and Europe, North America. So then we said that let's look at TMPRSS2. And we started looking at what are called EQTLs. Now, if you didn't know what EQTLs are, these are expression quantitative trait loci. These are really related to, these are variants that, genomic variants that actually impinge or impact on the quantity of the protein that it will produce, that particular gene produce. Not every polymorphism, not every mutation will impact on the level, the quantity of the protein that that particular gene produces. But these EQTLs are the ones that are of absolute importance, we believe, because it controls, it impacts on the quantity of protein or whatever that particular gene produces. So what we tried to do is to look at EQTLs, and we looked at EQTLs of TMPRSS2. And remember, TMPRSS2 is an enabler of the virus to get into the host cell. I'm going to cut a long story short. What we found was that indeed there is an EQTL, which is very interesting. The TMPRSS2 gene, if you look at it from 5' to 3', there is another adjacent gene which is oriented in the opposite direction. That's called MX1. And this particular EQTL is shared between MX1 and TMPRSS2. And MX1 also plays a role in enabling the virus to enter the host cell. And this particular EQTL, the polymorphism that we were looking at, is a polymorphism where there is a deletion of one nucleotide C. So it's like the normal individuals would have C at a particular position. The mutant individuals at that particular position would have a deletion of that C, would not have that particular C. So C and deletion of C, that's the polymorphism that we're looking at. And again, I'm not going to get into the gory details of it. My time will run out if I get into the gory details. So essentially what happens is that this particular del C polymorphism or deletion provides an added advantage for the virus to enter the host cell, and this is present in A2A. So the A2A is a double whammy. It's able to attach itself and is also able to get itself better into the host cell. But this del C, this is a human polymorphism. This del C is very different in terms of its frequency in Europe, North America versus East Asia. So the difference in the frequency of this particular mutation, the del C mutation, in East Asia and Europe, North America, so we believe, and its role and the role of this particular deletion in the entry of the virus into the host cell and its ability to, get into the host cell and infect more people. We believe that this differential between East Asia and Europe, North America virus to enter humans who reside in East Asia compared to the humans who reside in North America. So what we were up until this time, what we've been able to infer, or we've been able to infer, is we've gained insights as to why this one particular subtype has been able to spread throughout the world. This subtype, the 8A, that's facilitated by a specific mutation, which is aspartic acid glycine mutation that is in the virus, has given rise to this particular subtype called 8A. Then we looked at, so that's a universal spread. Then we looked at this difference in geographical, in these two geographies, East Asia versus continental Europe and continental North America. And we were able to understand variation, or at least pose a model that this is probably the reason why there is this differences in frequencies that we observe is because of this variation in a EQTL called that deletes a particular C nucleotide in the genome of the humans. So far, so good. So we've been able to explain spread and also differential spread. The last that I actually want to mention, I want to leave some time for questions as well. The last point that I want to mention is about the vaccine. In our ability to find a vaccine, there are various kinds of things that we must consider. One is that the vaccine, of course, we want it to be efficacious, meaning that it must provide us with protection against infection. That's the best vaccine that we would like, because then the virus won't enter. But there's also another kind of vaccine that's not too bad. Even if we are infected, we don't develop clinical symptoms, and therefore we don't die. Even that's protection. So you can think of vaccine in two ways. One is retarding the infection of the human host by the virus itself. That's one level of protection, and that's the best level of protection. You're closing the door on the virus, closing the main entrance door of the virus. But should the virus enter the main door, if you can close your bedroom door, then you'd be better off, because the virus won't get into your host cell and start reproducing there. So, and create other kinds of problems, such as clinical problems. So it protects you from clinical problems, therefore you don't die. So these are protection at two different levels. Of course, most people think, or most people have been trying to find a vaccine that will protect people against the infection. Nobody is sure whether this is quite possible, but I do want to spend the next few minutes on is to explain to you the phases that one needs to go through. We like it to be efficacious, but that efficacy doesn't come through so easily. So once you have a candidate vaccine, you believe that this is going to work. First of all, you have, even before you say that this is going to work on humans, you first have to take care of safety first. So it may be efficacious, but it can kill a large number of people, and you don't want such a vaccine in the market. As a matter of fact, there are many instances when a drug, which many humans are being benefited from, has killed a few people, and the drug has been withdrawn from the market. So do no harm is the first thing that you want to, that's the first basic tenet. So the first thing that if you have a candidate vaccine, the first thing that you do is to get hold of a few volunteers. The number is usually small, maybe 30, maybe 50. That's the kind of numbers that one looks at. Those volunteers are provided with this particular vaccine. And you look at safety. You look at adverse events in that particular individual for a period of time. And if there are no adverse events, then you say that my phase 1 trial has succeeded. The next phase of the trial is to look at efficacy. And the efficacy is usually seen initially in a small number of individuals. And of course, you're looking at a drug or a vaccine. It may be efficacious at one dose level. It may not be efficacious at a lower dose level. Or at a higher dose level, it might cause harm. It might lead to some adverse events. So what one does is to take, in the phase 2 trial, which comprises a larger number of people, you usually administer the drug at different dose levels, and you look at efficacy and also keep note of adverse events, if any. And that usually takes a longer period of time because you're looking at efficacy, so you also have to have a bunch of people, who may not be given the drug, etc. Small number of people. So that also takes a little bit of time. It takes about 6 months or so. And then finally, even before it comes to the market, you have to do a very large, what's called a phase 3 trial. So phase 3 trial is having found out that the vaccine does not cause any adverse events in phase 1, having found out the dose level at which the vaccine seems to be efficacious in the sense that it either elicits an appropriate immune response, it either does not allow the virus to enter, or even if the virus enters, it does not precipitate clinical symptoms. So you raise yourself to phase 3 clinical trial, and in phase 3 clinical trial, you recruit of the order of 30,000 people, and not just from one small region, you recruit them from all over the world because human genomes differ, their exposures differ, and because of differences in exposure to previous viruses, other bacteria, etc., their baseline immune levels may be low. And so you do this across the world as much as possible, spread out or recruit volunteers from various parts of the world. But the numbers that one looks at are of the order of between 10,000 and 30,000. So that's the kind of numbers that we look at. There are two vaccines with respect to SARS-CoV-2 that are near there, almost there, and phase 3 trials are starting. So the first one is the most close, that is past phase 1 and phase 2 and has come to phase 3. Phase 3 trials may have started just 2-3 days back. This is the final stages. This is the vaccine that's discovered, invented. What's the word? I don't know. Whatever is a vaccine that was invented by Oxford. University of Oxford in collaboration with AstraZeneca, which is a large pharmaceutical company. So Oxford AstraZeneca is now getting into their phase 3 clinical trials, or the final stage of the clinical trials, and they have recruited thousands of people in Brazil, South Africa, UK, and they will soon, or may have already recruited volunteers from the US. So this particular vaccine, the Oxford AstraZeneca vaccine, is a chimpanzee adenovirus. It's a common cold virus that infects the chimpanzees. Taken this chimpanzee adenovirus, inserted from the surface protein of SARS-CoV-2. Remember, the surface protein is very important because first player that was enhances itself, human host cell. So they've taken the monkey adenovirus, the chimpanzee adenovirus, and inserted genetic material from the surface spike protein of SARS-CoV-2, such that it elicits immune response. It's a benign adenovirus, made benign adenovirus with the appropriate SARS protein that does not cause an infection, but tricks the immune system. The immune system recognizes this as an enemy, and therefore it produces an immune response. But that immune response, is good, because when the true SARS-CoV-2, the vaccine, so the vaccine is like a proxy. Vaccine will elicit immune response. So when the true SARS-CoV-2 actually infects, the immune system is already primed to fight back. This particular adenovirus emits both antibodies. It elicits neutralizing antibody, as a matter of fact, that kills the virus, and also it elicits high level of killer T cells. The killer T cells are a type of white blood cells that helps the immune system destroy the infection. So it stimulates both antibodies and enhances killer T cells. So it seems like all that Jonas Salk, who was the inventor of the polio vaccine, the Salk vaccine doesn't actually stop infection, but it prevents disease from being. It prevents disease, which means that the child doesn't get crippled. And before the Salk vaccine was invented, millions of people have been crippled by polio. So the polio vaccine is a good example where the infection is not stopped, but the disease is stopped. But in this particular case, Oxford AstraZeneca, one believes that it is going to stop the infection because it's producing neutralizing antibodies and a large number of killer T cells. So this is going into phase 3 clinical trials. They've recruited 30,000 people from all over the world, and we hope that we will have the results. They are hopeful that they will have the results by about September, and within the year they'll be able to market their vaccine. They are so hopeful that they have also enlisted a large number of vaccine manufacturers, such that as the clinical trial is over, the phase 3 trial is over, and they are expecting good results from the phase 3 trials, they will be able to produce billions of doses of this vaccine throughout the world using these various companies that they have already enlisted. There is also another candidate vaccine that's very close. I believe it's not as close as the Oxford AstraZeneca vaccine. That's from Moderna. Moderna is a US-based company, and their construct is also very different. Like I said, that this is a positive stranded RNA virus. So what they have done is they have taken a cocktail of mRNAs of different proteins from the virus, and they have put that in a plasmid. The plasmid is able to self-replicate, and therefore multiple messenger RNAs. So as opposed to only one protein, which is the surface protein of the Oxford coronavirus vaccine, the Moderna vaccine has a cocktail of multiple messenger RNAs. So if it is successful, it may actually be more efficacious. However, must tell you that the phase 2 trial of the Moderna virus, Moderna vaccine, actually produced neutralizing antibodies, but it also produced some side effects in humans. A small number of humans, but it also produced side effects, while the Oxford vaccine did not. I don't hold any stocks and shares with either the AstraZeneca or with Moderna. So don't misinterpret me. I'm not saying that I'm backing the Oxford vaccine, but these are facts that the Oxford vaccine is probably more close compared to the Moderna vaccine, but the Moderna vaccine is also a great candidate. And these are the two that are sort of on the front of the race. And there are, I'm told that there are 263 vaccines that are being tried all over the world, 2 of them in India. But again, everybody else is far behind compared to the Oxford AstraZeneca vaccine and the Moderna vaccine. All right, so this is time for me to stop. I know that I haven't left any time for you to ask me questions, but if you have the time, I don't have a dearth of time, so I'm happy to ask questions. And I hope that I've been able to tell you about, the evolution, the spread, the biology of the wide spreads, and what's going on in the field of vaccine development, and that the vaccine is our only savior. Because at this time, otherwise it's just going to be the natural spread, and it is going to spread because it doesn't kill many people. So it does not. If it did kill many people, it would be self-contained, because people who were infected would die, and with their death, the virus also would die. But this is not happening here because it's a kind virus. It is spreading very well. So in order to avoid the spread, the only way to do it is to find a vaccine that will protect people from being infected, or at least protect people from coming down with clinical symptoms, and therefore protect them from death. Thank you very much for your attention, and I'm happy to take questions.
Such a wonderful talk, such a wonderful talk. And there are lots and lots of questions. Shaunak.
Yeah, thank you, Professor Majumder, for such an insightful talk on unraveling the biology of SARS-CoV-2, including the evolution of this deadly virus and its different subtypes. It's a pleasure to listen to all of them. We have received many questions, and it will take the whole day to answer all of them, but I'll try our best to discuss most of it. Many questions are on the same line, so I'll try to club them together for you, Professor Majumder. I'll try and read them here, and if you don't mind answering them. So question we received is, how many clades by subtypes are present in India currently?
Again, the number of sequences is very, very small that we found in India. And as a matter of fact, based on some small number of sequences, lots of tall inferences are being drawn to my consternation. So the answer to your question is we do find a dominance of A2A subtype, but we can't yet estimate the number of subtypes because sequences are being deposited in small numbers. I'm told that today's what, Sunday, day before yesterday, 1,014 sequences got deposited in GIS AID from India, and the effort was led by the National Institute of Biomedical Genomics. I have not had time to analyze that data, but next week is the whole plan of analyzing the Indian data. Right now from India, there are about 2,500 sequences. All over the world, there are now about 40,000 sequences. So you can imagine that our ability to robustly estimate the number of subtypes from the small numbers of data from India is limited by the fact that the number of sequences is small.
Okay, thank you. And as you have said that being an RNA virus, this virus has a huge potential to get mutated. And one of the, from the sequencing result, you've seen the D614G mutation. So does this mutation actually impacts the pathogenicity, and is this a roadblock to the generation of an effective vaccine?
By pathogenicity, you mean virulence. If that's what is meant, I should have mentioned, I did not mention, for all we can tell, of these 11 subtypes that there are of this human SARS-CoV-2, we do not find any particular subtype that's more virulent than the other. In other words, all of these types are actually quite kind, and there's no special virulence attached to any of these mutations. So the answer is that with respect to virulence, these different types are by and large the same, and it is not so virulent, and it's not so virulent. But this D614G has helped the particular subtype spread because of the biology that I explained to you.
Okay, thank you. And on the same line, we have received another question: is what is the frequency of the mutations or the mutation rate of the novel coronavirus now?
The mutation rate, like I said, it has a proofreading ability. Can't give you numbers. I don't remember the numbers top of my head. I do have them written down somewhere. The mutation rate is, most viruses have a very large mutation rate, very high mutation rate, of order of 10 to the power of -4. Just contrasting this with humans, it's like 10 to the power of -9 per nucleotide per year. Viruses is 10 to the minus, so it's 5 or 6 orders of magnitude higher. Standard viruses. This one is about 3 orders of magnitude higher. In other words, that it mutates slower than the influenza virus, for example, because of its proofreading ability. I also forgot to mention that the previous question asked whether that's a major problem with respect to vaccines. I think it's a boon, because if we find one particular subtype, then we don't have to deal with multiple subtypes, but we have to focus on this particular subtype, the D614G, and we have to address the G mutation as well, in addition to the D mutation. So in some ways, dominance of one particular subtype really helps find a vaccine.
Okay, thank you. We have received a lot of questions, and I think it's partly because of how it has been proved in the media that this virus could be genetically modified and coming up from a lab. But the most plausible hypothesis is with the sequence conservation that the SARS-CoV-2 actually evolved from a bat coronavirus with an intermediate pangolin. So going back to the conserved D614 mutation, which is present in the spike protein of bat and the pangolin, what selection pressure actually led to the mutation of this wild protein in the human cells?
No, selection pressure does not. Selection pressure plays no role in creating a mutation. The mutation gets created, and then Darwinian selection takes place. So there's no pressure that creates a mutation. Mutations are random events. And please don't get this wrong. In evolution, mutations happen first, and then the mutant, whether it's going to spread in the population, depends on the selective advantage or disadvantage that this mutation carries.
Okay. I'll go on to some questions on infections. What are the possibilities of reinfection by SARS-CoV-2 And if this is possible to
this is a very interesting question. All of us are actually grappling with this because as the site of the vaccine is coming closer and closer, how much protection or how long, what would be the period of protection that a vaccine can provide? We have no clue. As a matter of fact, because the virus has spread so quickly, everybody has been swamped with containing the spread of the virus, trying to see lockdowns and all of this. This is all to contain spread of the virus. So everybody has been just completely overwhelmed. There's no proper clinical data on reinfections. So we don't even know how often reinfections are taking place, what may be the period of reinfection. Absolutely no data, but this is a great question and we're all worried about it, especially in the context of the efficacy of vaccines, not the efficacy, the period of time during which they will remain efficacious to protect us.
Okay, thank you. We have another question. How can SARS-CoV-2 be airborne with respect to the World Health Organization as it settles down very early? Can CRISPR-Cas9 system be employed to destroy the viral particles?
Okay, the first question first. Yes, the virus primarily gets into the droplets. Of course, you know why. It spreads. It's a droplet spread, and it's not like it's not airborne in the sense that it is devoid of the droplet. It is inside the droplet. Now, droplets can be as small as aerosols, which can travel. So the aerosols carry the virus inside. The virus is really tiny, microns in diameter, and even the aerosols can contain some number of particles of the virus. The droplets can contain larger number of particles of the virus. So even if it is airborne, since the number of virus particles within the aerosol is small, is tiny, not very large. Even if somebody inhales the aerosols, the belief is that it's not the person may get infected, but may not elicit as much of clinical symptoms, may remain asymptomatic. While if you inhale a droplet or a few droplets, then of course you are inhaling a larger number of viruses, and therefore your viral load will. Then herd immunity is the only way to do it. Otherwise, we just don't have any choice. And so herd immunity, attainment of herd immunity will take a long time because for it to be effective, 50% is reasonably effective. 50% of the population herd immune is reasonably effective, but about two-thirds. If about a third of the population is herd immune, then you're sure that virus will not spread. And so herd immunity is the only way to do it, either naturally or artificially by a vaccine.
Thank you. And I'll go back to your initial first part of your talk.
And this came to us through this operation of A2A, differential spread of A2A in Europe, North America versus East Asia. And that's how we got into that problem, or got into looking at that problem. And we have figured out a biological answer to that. And the answer is that it is because of the genes and human SARS-CoV-2 interaction.
Okay. And I'll take a couple of more questions, like 2 or 3 more questions, if that's okay. So you got a question on, considering the virus's ability to mutate and the presence of asymptomatic patients, and also recent research suggesting placental transmission, is there a scientific possibility that the virus may remain undetected in a host and pass on to subsequent human generations and remaining with us forever?
Well, again, first of all, first things first, proximal question first. Let me give the answer to the proximal question. Yes, there are many asymptomatic carriers, and they
I think we are facing some technical problems with Professor Majumder.
spreading the infection will be transgenerational transmission stage, but it is possible that there will be transgenerational transmission. I think only time will tell. Again, all of these questions are so very important, but nobody has the time to look at these questions in any great detail. I myself have been trying to, get some work done on this, but the hospitals are just completely overwhelmed. They will not be able to collect additional data that's required for research. So that's the problem. So I think what we need to do is to wait for such time when herd immunity becomes a little bit more, attains a little bit higher frequency, and then the hospital load will come down a little bit, and then we'll be able to collect these data on these kinds of questions, to answer these kinds of questions.
And with the same question on the asymptomatic patients, so this question, if a person is asymptomatic, will he require vaccination in future?
Will he require?
Vaccination.
If the person is, if the person has elicited immune response, and that can be seen through neutralizing antibodies or IgG, whatever, then we wouldn't need a vaccination. But are you going to first test and then vaccinate? Probably not. So you just vaccinate everybody. If anybody who is not shown clinical symptoms will be vaccinated, I believe. But a better strategy would be to first find out whether the person has elicited any immune response to SARS-CoV-2 and then vaccinate. But then, some of these immune responses are non-specific. So if you have looked at IgG, for example, how do you know that this IgG is because of infection by SARS-CoV-2 and not by some other concomitant infection? So there are these issues, but if we are able to provide to billions of people, billions of doses of vaccines, and it can be made available cheap, then I think irrespective of asymptomatic previous infection, just get yourself vaccinated. I think that's the best.
Okay, thank you, Professor Majumder. And we have another question on, is there any correlation between a particular blood group and the prevalence of the disease?
Oh, well, yes, as you had seen, I know where this question is coming from. There was, I think it was the B blood group, right? Association, the B blood group were protected. That was on a sample of size, I forget what, but that study was enlarged to a larger sample size, and that particular association vanished. So we do not have any convincing evidence that blood group has anything to do with infection, susceptibility to SARS-CoV-2.
Okay, I think we'll take the last question today. According to latest studies, COVID patients showed neurological disorder. So is the viral virus able to bypass blood-brain barrier or any other possible mechanism?
Well, neurological doesn't always have to be blood-brain barrier. There are neurons elsewhere as well. So again, the point is that some I don't have the time to explain in any great detail. So a single cell map of AC2 has been done. So the question was, this virus, does it only affect or anchor to type 2 pneumocytes, or can it also anchor to other cells in the human body? And if so, then these other cells must also express ACE2, because this spike protein will go and bind to ACE2. So a single cell analysis of the whole human body or multiple human organs have been done. And we know now which organs express, cells of which organs express ACE2, and there are multiple organs that express, cells in which express ACE2, and therefore we know that the neurons or neurological problems arise. We know people lose sense of smell and taste, even though that seems to be transient. And we know that people develop gangrene. So I know, very sadly, mother of a student of mine, student of ours who is had to get a leg amputated because she had SARS-CoV-2 and developed gangrene. And so it impacts on various organs, impacts on various physiological functions, including blood flow. So it is, and it's all most likely related to expression of ACE2 on the surface of the cells in those organs.
Okay, and I'll have just one question. Last is, what is the basis of being asymptomatic versus asymptomatic carriers? Like, do you see there is a difference in the mutations that makes this difference, or is just the difference in the human immune response?
Good question, because but no answer yet. No, we don't have an answer. My own belief is that it will be in terms of human immune response. There's another very interesting thing that's been seen now, that people who live in slums, they seem to be protected better than people who live in, urban housing or other places. They're being in Dharavi is a good example of all of that. And that essentially says that poor hygiene keeps the immune system perked up, and therefore the chance of infection even by a virus is less. Now, does that mean that you have to live in unhygienic places to not get infected? But this is just an observation. So my own belief is that it's depends on the immune system and maybe other host genes that we still don't know. So susceptibility to clinical symptoms and susceptibility to infection may be two different ball games altogether.
Thank you very much, Professor Majumder, for that talk and for taking the time to answer all these questions. We do apologize. We have received a lot of questions. It's not possible to answer all of them in the short span of time. With that note, I now request Professor NIBMG to express a vote of thanks and close to the session.
Thank you, Shaunak. You have done a wonderful job. And thank you so much, Professor Majumder. As multiple narratives spin around COVID-19 and we live in terror, and there are discussions and counter discussions on science and pseudoscience and perceived science and infodemics literally drowning us. I think today's talk, as a thunderstorm rages outside Kolkata, people from all over the world are listening to your talk, and it was so enriching and so reassuring. So let us hope that the human resilience and the human mind will win over whatever obstacles are in our path. And thank you so much for a very reasonable. And one thing I must mention, usually we have speakers using slides and videos and animations, and when I asked you, you said I will just be talking. But then from where the last almost 1.5 hours went, we do not know. We were so mesmerized with your talk, and you enriched us so much, very matter-of-factly, very in such an uncluttered manner. I think that is probably the greatest take-home message from this talk. So thank you so much, and thank you to the team. I will be failing in my duties if I do not acknowledge Shaunak, whose idea this was, and how nicely he has hosted it. I remember when he was an MSc student in 2012, and from there he has become so self-poised and confident. It really makes me proud. There's Dushant, there's Sanjana from Louisiana, and Sundaram from Delhi, and Nondita Shinjini, and Pinku Haldar, who has designed our logo and is the main person behind all the intricate technologies of the projection and everything, and of course Pranab. I hope I have not missed anyone, and Rupkatha. So thank you so much all. একটা বৃষ্টি ভেজা সন্ধ্যায় আমরা এত চমৎকার কিছু কথা দিয়ে মন কে সিঞ্চিত করলাম, ঋদ্ধ হলাম। Thank you so much. Stay safe, everyone, and we will see you on the other side of the pandemic, hopefully. Thank you.
Thank you very much.
Thank you, everyone. Goodbye.
Bye.
Okay, thank you, sir. Thank you, organizers.